I had a weird dream last night that half stars that I was trying to eat.
That's fun. You have to. Alex Skarsgård.
It's like, I don't know, I'm pretty tolerant.
Okay, welcome everyone. How are we all doing? Good,
You're
super.
proud?
Um, remember we have an exam on Friday, right? Quiz. Okay, so weekly quizzes, so keep up with it. There will be a practice quiz that will release, essentially right before the regular exam. Okay, and the goal, as usual, is the practice quiz is going to be quite representative of the actual graded quiz. Any questions on that? Okay, pretty straightforward. Okay, so topic for today is heme synthesis and heme degradation. We're going to kind of gloss over heme function because we've talked about that in other contexts. The pathologies most of them are poryphyrias, and they're a little bit difficult to distinguish one from the other, although I'll give you some strategies to do that. So don't get too intimidated by it. On a test question, you're not going to have to distinguish porphyria cutanea tarda from acute intermittent porphyria necessarily, but you'll have to identify a porphyria and distinguish it from a urea cycle disorder or something like that. Okay, so it's a little tricky, but try to focus on the big picture. Okay, so before we get started on the heme synthesis, why don't we circle back to the case that we did not get to yesterday. So this was a young female baby who was born with jaundice and anemia like at birth. And so she had low hemoglobin and low hematocrit, so that's the anemia. She had a high reticulocyte count, so that's indicating that the bone marrow is responding. So she's trying to compensate for the anemia by increasing the rate of red blood cell production. So let's see what else. Oh yeah, and elevated bilirubin as well. Have you gone through Dr. Shaw's material on what a red blood cell smear would look like? This isn't like a good quality image, but what are your impressions on the appearance of the red blood cells? Normal or abnormal?
Abnormal.
Okay, and why do you say abnormal?
It looks like a lot of reticulocytes and like maybe kind of misshapen ones. Like there's like almost two that are together. It looks like in spots. They're not round.
Yeah, that's kind of like I'm not a histologist and certainly not a hematologist. So when I look at the red blood cell, they're supposed to all be the same, and these are not all the same, right? Fair enough. Okay, so there's something wrong here. If you look closer, you can see the membranes are a little ragged, suggesting maybe a hemolysis event. Okay, so this young child had phototherapy. So elevated bilirubin and jaundice is not uncommon in a newborn. And we'll talk about that a little bit in today's lecture. And phototherapy is a common treatment for that. The UV light just breaks down the bilirubin, as an alternative route for the breakdown. So phototherapy was initiated, and let's see, the green line here is the bilirubin, and that decreases. She also had 2 transfusions, and then that corrected her anemia. The hemoglobin is the red line here. And then the nucleated red blood cells, as the anemia is corrected, those decrease as well. Okay, so that was good. However, over time, and then we get kind of a jump here into the weeks level, her anemia is returning. Okay, so she got another transfusion here, and then that corrected the anemia again. However, it didn't correct it on a durable nature. In fact, she had to have, over the first 2 years of her life, multiple transfusion events to correct anemia. Any, any thoughts on a potential complication for having this many transfusions this rapidly? No. So we're going to talk about iron metabolism next week, and you could potentially get hemochromatosis or an iron overload condition from having these multiple transfusions, okay? Because, well, I don't want to ruin the story for you next week, but basically your iron amount in your body is regulated by uptake of dietary iron. Okay, so with this transfusion, you're bypassing that regulation and giving the patient excess iron in the arm into the vein, right? So you get accumulation of iron and no real way to get rid of it because the normal regulation is all at the level of uptake. Okay, anyway, so something to think about. In this patient, she had a splenectomy, so removed the spleen, and then that was a more durable cure for her anemia. Okay. Thoughts? All right. This is an example of a red blood cell enzymopathy. So you've talked about some hemoglobinopathies of red blood cells, right? Like sickle cell disease, for example. This is an enzymopathy of red blood cells. One of the most common one is glucose 6-phosphate dehydrogenase deficiency. In this case, the patient had pyruvate kinase deficiency. I think we talked about pyruvate kinase in foundations, where remember there's the M1 isoform, and then in cancer, the cancer cells will revert to the embryonic M2 isoform, if you remember that story. Okay, so this is a totally different gene that is encoding both the liver isoform and the blood cell, the erythrocyte isoform of pyruvate kinase. And in this patient and in this cohort, this family that's this condition was identified in, they have decreased function, or excuse me, inherited mutations that cause a decreased function in the erythrocytes isoform of the pyruvate kinase gene. So the consequence here is that the cells have a failure to generate enough ATP to sustain their membranes, to sustain their cell membranes. The cells then dehydrate and they have a difficult time getting through the capillaries and the circulation, and that gives them that kind of ragged appearance in the smear that we saw. Now, the presentation clinically, it's variable depending on how much of the activity of the pyruvate kinase is there, and that's going to be dependent on which particular mutation you have. So you can have just a asymptomatic compensated anemia or a more severe condition, as was shown by our child. Just as a kind of interesting feature of this condition, with the pyruvate kinase activity low, you get a buildup of all the intermediates of glycolysis, and that causes a diversion of the substrates into the Rappaport-von Luberen shunt and generates more 2,3-bisphosphoglycerate, and that can help to not compensate for the anemia so much as, I guess, increase the efficiency of the red blood cells that are there. Okay. Okay. Any questions about this condition? And this comes from a case report 10 years ago or so from the U.S. here. Okay, so that's, I guess, the second most common of the red blood cell enzymopathies. Glucose 6-phosphate dehydrogenase deficiency is the most common one. Should we try a few quick clicker slides? warms my heart. Okay, so glycine is one of the two substrates for heme synthesis. That's why I have that on here. You want to consider the stoichiometry of hemoglobin is as a heterotetramer. So each of the 4 subunits has one heme molecule, and each of those heme molecules can bind one molecule of oxygen. Okay, now cobalamin, of course, is important for methionine metabolism, right? But pyridoxal phosphate, that's kind of universally used by enzymes that do all sorts of different diverse reactions using amino acids for substrates. Okay, so pyridoxal phosphate or vitamin B6 deficiency, the main symptoms are neurological, like peripheral neuropathy and seizures and et cetera. But what's another symptom of vitamin B6 deficiency? And you could just look at the academic calendar and see what we're talking about this next few weeks and probably guess. But well, the exam soft is being kind of slow. Yeah, so vitamin B6, one of the other symptoms is anemia, and that's because vitamin B6 is required by the key regulatory enzyme or the rate limiting step of heme synthesis. Okay, got a few other questions here. And these questions seem kind of random, but they're all kind of, you know, somewhat leading to material that we'll talk about. And the TCA cycle enzyme here isn't so important, but succinyl-CoA, in addition to glycine, succinyl-CoA is the other substrate for heme synthesis. And maybe you want to review the Stanford pathway here. Correct answer is the thiokinase, actually. So, okay. All right, 3 more questions here. Okay, so Lineweaver-Burke plots is a way to express Michaelis-Menten kinetic data. Great, very good. And we'll see shortly how lead affects the kinetics of aminolevulinic acid dehydratase and causes anemia as a result. Okay, so we've seen, you know, plenty of examples of this before, and just want to bring this up to illustrate that this is how heme synthesis works. So the product of heme synthesis is the molecule heme, and it acts as an inhibitor on the first enzyme that commits substrates to heme synthesis, the delta aminolevulinic acid synthase. Okay, final question here. Is this something that Dr. Cormier talked about early in your career as med students? Okay, oh, pretty good. So it's like got a majority of you picked the GU on the 5' end and the AG on the 3' end. Great job. Okay, so we'll come back to that, circle back to that. All right, so topic for today is heme synthesis, and this is in the desert southwest of our Stanford map of human metabolism, just southwest of the TCA cycle. You've got the synthesis down here, and the degradation of heme is over here. So the high-level view of heme synthesis, the two substrates are succinyl-CoA and glycine. Those are the only two things that you need to make heme within our body. Oh, and iron, I'm sorry. Iron, of course, is a key component as well. So disorders of heme synthesis generally cause porphyrias, or disorders that are caused by accumulation of the intermediates of heme synthesis. Disorders of heme breakdown lead to hyperbilirubinemia and jaundice. Okay, that's kind of high level view. Oh yeah, and then kernicterus, this is referring to brain damage or neurological damage due to hyperbilirubinemia. Okay, so what is heme? Heme is a tetrapyrrole. So the structure of a pyrrole ring is 4 carbons joined by an X group into this pentameric ring. In the case of heme, the X is a nitrogen group. So you have these 4 rings, here, 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 and here, and each of those rings has a side chain on it, and the side chains are methyls, propionic acid, and then vinyl groups here. And at the center of the ring is a molecule of ferrous iron, of +2 iron. Okay, so that's the basic structure of heme, and it makes, in 3-dimensional space, it makes a planar structure. Okay, so this is all kind of, as it is in 2 dimensions here. Where it functions, now heme has a lot of functions within our body. We're going to focus on its function in hemoglobin because of that's the topic of the course that you're in right now. But heme is important for some enzymes on the electron transport chain. Cytochrome P450 enzymes use heme. Myoglobin in muscle uses heme. So there's a lot of kind of non-hemoglobin uses of heme within our body. But red blood cells are using the most heme, and because red blood cells turn over so rapidly, that's the greatest kind of reason that we do heme synthesis, is to support red blood cell synthesis and turnover. Okay, so the heme is coordinated by the amino acids in hemoglobin, and those propionic and vinyl, or propionic acid and vinyl and methyl side chains, those project out of the heme molecule and interact with atoms on the side chains of the amino acids in the globin proteins to stabilize it. And each of the monomers of hemoglobin can bind 1 heme, and then that results in 4 oxygen binding sites for any 1 molecule of hemoglobin. Okay. Oh yeah, and the major sites of heme synthesis, so erythroid precursor cells in the bone marrow, and that's supporting erythroid cells, of course, and then the liver also makes heme, and the liver is kind of making heme to support the other diverse functions of heme within our body. Okay, so there are 8 enzymes that are required to synthesize heme. So the substrates are succinyl-CoA and glycine, and then ferrous iron at the last step. So the first steps and the last steps take place in the mitochondria, and then the intermediate steps take place in the cytoplasm. So there are these transport processes for the intermediates to go into and out of the mitochondria. As I mentioned in the previous slide, the main sites of heme synthesis are in erythroid precursors and in the liver. And the enzymatic steps are the same in those 2 compartments, right? So the steps are as shown here in the same order in the liver and in the red blood cells, but there are some different isoforms of the enzymes that are carrying out the steps. And the one that we'll focus on is that first enzyme, the delta aminolevulinic acid synthase, and it's regulated in kind of subtly different ways in the liver and in the red blood cell precursors. Okay, so just to look at the synthesis of heme from a structural standpoint, and I'll kind of try to go through this somewhat quickly. Okay, so the key rate limiting step and the key regulated step is the delta aminolevulinic acid synthase, and this is negatively regulated by heme. And then it's also regulated at the expression level in different ways in erythroid precursors and in hepatocytes. So the enzymatic activity of delta aminolevulinic acid synthase is to take succinyl-CoA and glycine and to decarboxylate the succinyl-CoA and then remove the coenzyme A part of it and join it with glycine to make aminolevulinic acid. So aminolevulinic acid, this is an amino acid, but it's not like an amino acid that would be used to make proteins, of course. Okay, so the aminolevulinic acid, this then goes into the mitochondria, and it's a substrate for aminolevulinic acid dehydratase, and the dehydratase enzyme takes 2 aminolevulinic acids and condenses them together to make the first pyrrole ring structure, this ring structure. So after the first ring structure is made, porphobilinogen deaminase will remove this nitrogen as ammonia and then join 4 of those pyrrole rings together to make the first kind of complete structure of the heme, if you will. Let's see, the next step involves another dehydration reaction that will close the ring, and here we get the first porphyrinogen. So once you get the ring closed, we refer to these structures as porphyrinogens, and there are some symptoms of the different porphyrias that are particular to generation of things downstream of the porphyrinogens. Okay, so I'll elaborate on that later. Okay, so after the ring structure is closed, then the subsequent steps are modifying the side chains. So it's taking a couple of the propionic acids and decarboxylating them into the vinyl groups. Then it's taking some of the acetic acids and changing them into methyl groups through a decarboxylation reaction. Then in the in the penultimate step, we get the conversion of these bridges here that are linking the pyrrole rings. We get the oxidation of those, and then finally we get the addition of ferrous iron biferrochelatase to make the ultimate heme structure. Okay. Clear as mud? Okay. All right. So there it is, and I don't know. Do you need to memorize it? Do you need to be able to draw it on a blank piece of paper? Probably not. But there it is for your reference if you wanted to do that. Okay. Let's talk about, I guess, why don't we focus from here on in about the regulation of heme synthesis, and particularly the regulation of aminolevulinic acid synthase in hepatic tissue and in erythroid tissue. And then we'll focus on some disorders of heme synthesis, and hopefully it'll make it a little bit easier to look at the consequences of disorders at different steps of the pathway. Okay, so aminolevulinic acid synthase, this is the key regulatory step, and this is the regulatory or the rate limiting step for heme synthesis both in erythroid precursors and in hepatocytes and other tissues that synthesize heme. So the aminolevulinic acid synthase is under negative feedback regulation from heme, the final product of the pathway. So this is wonderful in healthy people that don't have any issues with the pathway. However, this negative feedback inhibition causes real problems if we have a block at some point in the pathway. Okay, because say if you lose the coproporphyrinogen oxidase enzyme, you won't get the heme produced, right? So you won't have negative regulation of the aminolevulinic acid synthase. That's going to be operating at full speed. Okay, so the symptoms then are not just going to be from the normal amount of the coproporphyrinogen accumulating, but because you've got unregulated substrates coming into the pathway, you're going to get a huge exacerbation of the amount of intermediates that build up. Is that clear? Okay, so that's a kind of a key feature that drives a lot of the porphyrias that we'll see. Okay, so why don't we look a little bit more closely at the aminolevulinic acid synthase enzyme. Aminolevulinic acid synthase, it is condensing succinyl-CoA and glycine in a decarboxylation reaction and using the energy of that high energy bond between the succinate and coenzyme A as the kind of energetic driver of the reaction. ALA synthase in both erythroid and hepatic cells requires pyridoxal phosphate, vitamin B6, because one of its substrates is an amino acid, so that makes sense, and its product is an amino acid. So the dependence of heme synthesis on pyridoxal phosphate is illustrated by this clinical case where a patient had a pancreoduodenectomy to remove cancer. So pancreas and part of the small intestine was removed, and that duodenum, that's the site where vitamin B6 is absorbed principally in the diet. So we had the pancreoduodenectomy, am I saying that right? Close there, right here. And the patient's hemoglobin had a steady decline until it's kind of crashed at this point, so it became pretty severely anemic. At this point, the vitamin B6 deficiency was recognized and oral pyridoxine supplements was initiated, and then you get a restoration of the hemoglobin levels. So vitamin B6 levels in the blood are, I think, shown on this line. There are some other interesting parameters that are measured here having to do with iron metabolism, so total iron and ferritin iron. So maybe just put a pin in this slide, and after our iron talk next week, maybe review some of this, and if you have questions about it, come and see me. Okay, so yeah, this just illustrating heme synthesis is dependent on vitamin B6, the basic point here. Okay, so that ALA synthase in both hepatic cells and in erythroid precursors is dependent on pyridoxal phosphate. However, the regulation of the expression of the two enzymes is different in the two different compartments. So ALAS1 is the kind of ubiquitously expressed aminolevulinic acid synthase gene, and that's responsible for liver production of heme and production of heme in all other tissues. And then ALAS2, this is the specific enzyme that is restricted to erythroid precursors. So this is the one that's dedicated towards making the heme that will wind up in hemoglobin. So they're both, the expression and activity of these enzymes is both regulated by heme, but in subtly different ways. So if we look at the ALAS1 expression, the ALAS1, so this is encoded from a totally different gene than the ALS2. And the regulation of ALAS1, I guess the things that are positively regulating it are tissue energy starvation and various forms of low energy, such as the high ratio of NAD plus to NADH in the cell. That's going to activate the PGC1 alpha transcription factor and then increase the transcription of the ALAS1 gene. And if you think about the function of heme in a lot of other tissues besides the red blood cells, heme is a way that we can generate energy from electron transport chain, for example. So it makes sense to couple the expression of heme to the energy status of the cell. Okay, so once the mRNA is translated, the regulation of ALAS1 expression by heme is at the level of degradation of the mRNA. So mRNA instability is promoted by heme, and so degradation of the mRNA is one way to choke off the, or to get that negative feedback regulation of heme. An additional way is to prevent the import of the ALAS1 protein into the mitochondria where it does its function. If you remember, so mitochondrial proteins that are encoded in the nucleus, they have a leader sequence of positively charged amino acids that plugs them into the mitochondria. So that's why we're calling this the pre-ALAS1. And then once it goes into the mitochondria, that little targeting sequence gets chopped off. So that step is inhibited in hepatic tissues by heme. Okay, so that's this case in the hepatic tissues or tissues that express ALAS1. ALAS2 is also regulated by heme, but it's in a subtly different way, or I guess it's in a less direct way, shall we say, and it's more due to the intracellular iron pool here. Okay, so in ALAS2, you have, oh, I'm sorry, I got ahead of myself here. So this is still, and this, I'm not sure why this is here. Is this slide in this position on yours? Okay. All right, let me go back to, well, way back to this. So that feedback inhibition idea and how that gets lost if you have a porphyria or a deficiency in one of the enzymes here. You can treat a porphyria by the addition of exogenous heme. So hemin is it's a formulation of heme that's, the details of it is subtly different than endogenous heme. Anyway, you can treat these conditions with heme, and that doesn't stop the block in the pathway, but it can diminish the exacerbation of the substrates going into the pathway. Okay, so all right, I apologize for getting that out of order. I'm not sure why that ended up there. Okay, where we were going was ALAS1 versus ALAS2 and the regulation. So ALAS1 expression and activity is directly regulated by heme. And in ALAS2, the regulation is through the intracellular iron pool. So in a red blood cell precursor, when we're making heme, that last step, the ferrochelatase enzyme, is consuming intracellular iron and depleting the intracellular iron pool. Okay, so that's shown here, right? So I guess it's not indicated here, but there's a ferrochelatase enzyme that's sucking up this ferrous iron and depleting the iron pool. So the regulation of ALAS2 expression is at the level of regulating the access of the mRNA, the ALS2 mRNA, to the ribosomes. So it's a regulation at the translational step of gene expression, and it's directly sensitive to the amount of iron within the cells. So I'm not sure if Dr. Cormier talked about iron regulatory proteins or iron response elements. Did he touch on this in gene regulation? Yes? Okay. All right, so hopefully this will be somewhat familiar to you. And this, we will circle back to this next week when we talk about iron metabolism and iron homeostasis. Because in addition to ALAS2, there are a number of other genes that are involved in iron in one way or the other that are regulated in the similar fashion to be sensitive to how much iron is within the cell. Okay, so with respect to the ALAS2 expression, once the transcript is generated, the ALAS2 transcript is generated, there are hairpin structures in the 5' end of the mRNA that are iron response elements or are called iron response elements because they bind to a protein called the iron response element binding protein. So when the iron response element binding protein binds to that hairpin, it prevents access to the ribosome and prevents translation of the enzyme. Okay, so it's in the absence of iron, you are not going to be making any ALAS2. And that is a good thing because heme synthesis requires iron, and if you don't have iron for that last step, you don't want to commit the enzymes to the earlier steps, right? So it's efficient. Okay, when iron is replete within the cell, iron will bind to those iron response elements binding proteins, and the iron binding changes the conformation of the proteins and releases the ALAS2 mRNA from the iron response element binding protein, and that relieves the translational inhibition, allows the ribosomes to access the message and to synthesize the ALAS2 protein. Okay? And then the ALAS2 protein then is imported into the mitochondria, and then it does its job of initiating heme synthesis. Okay, so this is the case with ALAS2. There are other genes that are regulated in this fashion, and there are other iron response element binding proteins that bind to the 3' end of messages in some genes. So we'll elaborate on this more next week when we talk about iron metabolism. Yes?
In the previous slides, the little green circle, that represents mitochondria, right?
This?
Yeah.
Yes, this is a mitochondria.
So is all of the heme made for the red blood cells when they're still, so erythroid cells are like pre-red blood cells, right? Okay.
Yes.
And then they kind of export their mitochondria, and they just have all the heme that they're going to have for their life until they die.
That is correct.
Okay.
Yes. Yeah, so when we talk about expression here, we're talking about in the bone marrow before they have pooped out their mitochondria and dissolved their nucleus. And, you know, that's a process that you're going to learn a lot about, I think, or if you haven't already, right? Okay. Okay, yeah, a good question. Now, once the hemoglobin is made, or once any enzyme in the red blood cell is made, whether it's a glycolysis enzyme or what have you, the mature red blood cell has no capacity to synthesize a new one to replace a defective enzyme. Okay, it just does its business until things crap out, and then it gets taken out by the macrophages of the reticuloendothelial system. Okay, let's see. All right, so are we clear on regulatory differences between ALAS2 and ALAS1. And, for the purposes of this class, we'll focus on ALAS2 because this is the hematology class. Okay. Are there questions on normal heme synthesis? Okay, so I'll talk about disorders of heme synthesis, and there are a couple. Well, there's a nutritional deficit that we talked about with the vitamin B6 deficiency causing anemia because of its importance in aminolevulinic acid synthase activity. There's also a toxin exposure aspect. So ALA dehydratase is one of the targets of lead, and anemia from lead poisoning is due to this inhibition of ALA dehydratase and ultimately heme synthesis. So there's a couple of kind of nutritional and exposure issues with heme synthesis. There are also many inborn errors of metabolism, and one of them results in an increase in activity of an enzyme, and that's this sideroblastic anemia. All of the other ones, that's the X-linked erythropoietic protoporphyria. All of the other ones can be caused by inherited mutations that cause a loss of function in the enzyme. They can also occur sporadically in the absence of any known mutations, and I'll provide an example and a mechanism of one of those later on in the class. Okay, so let's look at, well, we're generally going to go from north to south down the pathway here, and I'll try to hit kind of the pathologic importance at each step. So we'll start with the aminolevulinic acid synthase, and we already talked about vitamin B6, so I won't say anything more about that one. But I'll spend a moment here and talk about the sideroblastic anemia, which results from loss of function mutations in ALAS2, or in X-linked erythropoietic protoporphyria, which is one of these rare conditions that's caused by a hyperactivating mutation. Okay, so sideroblastic anemia, and X-linked because the ALAS2 gene is on the X chromosome. This is really, let's see, if we look at this pathway, this is the only one that is not going to have features of a porphyria. So the porphyria, the basic pathologies are going to result either from a buildup of aminolevulinic acid or from a buildup of the porphyrinogens or from both, okay? And if you don't have activity of the ALAS2, if you don't have aminolevulinic acid synthase, you're not going to produce aminolevulinic acid or the porphyrinogens, okay? So sideroblastic anemia is kind of a separate thing from the other pathologies that we'll discuss today. Okay, so with sideroblastic anemia, you get a anemia that is hypochromic and microcytic because of a lack of heme synthesis and then a lack of hemoglobin synthesis. So I'm not sure if Dr. Shaw or if maybe the pathologist talked about, like, if you look at, the red blood cell, it's got the dark red ring and then a pale pink center, right? And that dark red ring, normally it's going to be about a third of the or a third of the width of the cell. Has anyone told you that, or am I imagining that? Okay, you may hear that from someone shortly. You'll see there are a lot of cells here, like this one, where that dark red area is really thin. Okay, so that's like a cell that doesn't have a lot of hemoglobin in it. Okay, so with the X-linked sideroblastic anemia, you get this microcytic hypochromic anemia. And then if you look at red blood cell precursors in the bone marrow, you'll see that they're rich in iron. Now, if you do this Prussian blue cell staining of bone marrow, it's normal to see a few dots of iron, but if you have abundant iron in that, that could suggest either a hemochromatosis, iron overload, or some failure of these precursor cells to use iron. The ferrochelatase basically doesn't have any substrates in these cells, so the iron is accumulating to abnormally high amounts. Okay, so loss of ALAS2 activity mutations causes X-linked sideroblastic anemia. If you have a mutation that causes a hyperactivation of the ALAS2 gene product, that causes X-linked protoporphyria. And this is a porphyria, so it has the hallmark symptoms of skin blistering because of the accumulation. In this case, the accumulation is primarily this last one, the protoporphyrin. So with the X-linked protoporphyria, here you again don't get the heme produced, so you don't get the aminolevulinic acid synthase negative regulation, and then you get the hyperactivation mutation that's kind of greatly exacerbating the symptoms. Now, because with the X-linked protoporphyria, you don't have any other enzymatic defects in any of these other steps, the main thing that builds up is this final product, the protoporphyrin, because you're making those intermediates and exceeding the iron capacity of the cell or the iron stores of the cell. So this protoporphyrin is the one that really builds up. And in fact, the symptomatic profile and the biochemical profile of patients with the X-linked protoporphyria, gesundheit, that recapitulates people who have a defect in ferrochelatase, so that final step. Okay, if you have a defect here, of course, obviously you're going to build up the protoporphyrin as the major product, but that's also the case with the ALAS2 gain of function mutations. Okay. All right, so with our heme synthesis disorders, we've got the X-linked erythropoietic protoporphyria that's caused by a mutation that increases ALAS2 function, and then all of the other ones, sideroblastic anemia and downstream, are due to loss of function mutations. And yeah, so the main clinical features of the porphyrias are neuronal symptoms, and that can be both the central and peripheral neurological systems, as well as the GI neurological symptoms. So you can get nausea, vomiting, diarrhea, etc. So the GI and neurological symptoms are due to accumulation of aminolevulinic acid. Skin problems, and the major skin problem is blistering in areas of the skin that are exposed to sun. Those are caused by the accumulation of these porphyrinogens. Okay, so you can use just the symptom profile of a patient as to whether they have GI symptoms or skin symptoms or both to kind of somewhat deduce where in the pathway the issue is. Okay, and I think that is shown here. Porphyrias are generally divided into acute porphyrias and non-acute porphyrias. So the acute ones, these again have similar symptom profiles, but they are episodic and transient, and generally they're going to be brought on by some viral infection or drug exposure or toxin exposure that results in oxidative stress. And then this is in contrast with the non-acute porphyrias, which can, in the case of porphyria cutanea tarda, can be precipitated by oxidative stress, certainly as part of the mechanism, but it's more of a slow burn accumulation. You don't get the rapid exacerbation of the symptoms in an episodic fashion. Okay, why don't we stop there and resume our discussion of the porphyrias at 11 o'clock?
Like that hour 3 almost hits the hard. Yeah, let's walk.
Let us walk.
Erica, would you like to walk? Yeah, come walk.
There's a restaurant called Walk and Go. Oh, you can bring my.
Yeah, you walk and then go. There's no
Well,
tables.
There's tables.
To compare another country.
Yeah, he was really cool. He was pretty subjective to